So good afternoon, everybody, and welcome to the Institute for Quantitative Biomedicine seminar series. I'm Stephen Burley, the founding director of IQB, and very pleased to be hosting a friend and colleague, Dimitris Metaxas. But first, a few housekeeping announcements. We will be on hiatus for a couple of weeks. And then on the 20th of March, Harold Schwalbe, who's an expert in NMR studies of biological macromolecules, particularly RNA, and he'll be talking about the work that he and his team did looking at the SARS-CoV-2 genome. So he's speaking on March the 20th, and then on the 27th, Tanya Kortem will be visiting from UCSF. She's part of the Rosetta Mafia. So I hope you ensure you make time for her visit as well. I will eventually figure out how to use this this mouse wheel. The RCSB Protein Data Bank continues to recruit in terms of scientific software developers, recent graduates looking for gap year or gap half year experiences, and also undergraduate interns. So please look at our jobs page, www.rcsb.org/pages/jobs for more information. For those of you who are interested in understanding more about how the Protein Data Bank works, we have instituted a series of virtual office hours for this calendar year, and the next will occur on the 28th of March at 1 p.m. Eastern Time. And the focus will be Ask a Biocurator, and I'm grateful to both Brian Hudson and Ezra Pasak for agreeing to host this session. The IQB is a family that relies on the efforts of a number of people who work behind the scenes. Here are their pictures. Please be sure to be grateful if you see them in the hall. So with that, I'll brief introduction. I will actually move to introducing today's speaker, distinguished Professor Dimitris Metaxas of the Computer and Information Sciences Department here at Rutgers. Dimitris has a number of leadership roles here at the university and in a big NSF university industry collaboration center known as CARTA. He is a PhD graduate of the University of Toronto, where he worked in AI and machine learning. Though many of you may not appreciate this, but the University of Toronto was one of the world's leading institutions in AI and machine learning before it became fashionable, before it became faddy, and all the other institutions jumped on the bandwagon. The Canadian government continues to invest heavily in AI at the University of Toronto, and I'm sure that we'll continue to see major advances coming from there. Dimitris has worked across a whole host of different areas in applying AI and machine learning tools in different areas of science. And today we're going to hear about the work that he's been doing in cancer detection. He's one of the top people in the world in AI. He's in incredible demand as a speaker. And so I'm very grateful that you carved out the time to be here today. He has published more than 700 research articles, which is almost precisely double the number that I have published. Impressive, thank you very much. And graduated more than 65 PhD students, many of whom are working in prestigious academic and industry positions. His work has been funded over the years by NIH, NSF, AFOSR, ARO, DARPA, HS, ARPA, and the Office of Naval Research. Dimitris's work has received many Best Paper Awards over the years. He was a Fulbright Fellow in 1986, and he's a Fellow of the American Institute for Medical and Biological Engineers, a Fellow of the IEEE, and a Fellow of the MICCAI Society. He's also a key organizer of important meetings in AI, and will serve as the general chair for CVPR in 2026. So Dimitris, I'm very happy that you're here today, and I am looking forward to this seminar. I'm also looking forward to the next seminar that you'll give in. We'll probably do jointly in this room on our own work together. So thank you very much.
Thank you very much, Steve, for your introduction. I got, after Toronto, I was there for 10 years at UPenn, where I got trained into doing multidisciplinary research, which of course made a big difference. So when I came here, I started the Center for Computational Biomedicine, Imaging and Modeling, with the idea to foster collaboration so that we can do a lot of innovations in other fields, especially in medicine biology. So I'm glad I met Stefan, even though I was on the team to hire him. I met him several times, but it takes time these things to happen. So today I want to tell you about AI, right? So most of you know of AI, but what's happening now, there's a new term called AGI. How many of you have heard of this? It's artificial general intelligence. We're trying to be like humans. So I'll show you some breakthrough we had in the last few months, and I am submitting it actually next week to show you how this can be done so that you don't use them as black boxes, but they explain to you what they do. All right, so but let me start now with this research we've been doing on cancer detection. So the idea here is, sorry. Oh, it's not. Oh, yeah, right. Sorry about this. Right. So digital pathology, right? So it's a golden standard for measuring the originator of diseases, over its resolution, cell level morphology, shape, appearance. And you won't diagnose cancer, obesity, many type of disorders through images or other measurements. But here I'm going to focus primarily on images. I got introduced to pathology department at Penn. So I had a student actually around mid-90s when we started doing this in breast cancer and histopathology. And I've been interested since then for a long time in this topic. So what is happening, right? So, let me put this. All right. So, the whole spectrum, right? You have from genes, go to protein disorders, cancers, and then tumor. When I speak to computer scientists, of course, I have to explain a lot more. You guys know that way better than I do. So the question, why is not? Oh, okay. So you see a tumor, right? So you do lab tests, imaging tests, histopathology. Then you do surgery, radiotherapy, if you're dealing with to remove the cancer. Protein disorders, you have targeted therapy and through genetic tests, right? And then gene therapy if possible. So this is the whole spectrum, right? So you have data through the whole thing. So that my vision or my dream is, could at some point have this whole spectrum of data. So I can say, given, for example, the image here, what happens to the genes? You can learn correlations at some point, given data sets. So, and not only to distract, can do 3D structures, can do a lot of things. The problem, of course, that we used to face a lot, or still is, like, look how big is histopathology image. I mean, I still remember going to Penn to the histopathology department. These guys were killing their eyes, hours and hours, and they were making mistakes because when they get tired, they're humans. So, of course, what do we do? And if you zoom in, of course, you see this picture here where there's cancer all over the place. The biggest problem, from what I remember clinically, what I know clinically, is that how many types of cancer do you have? Because then you're in trouble, right? So if it's only one type of cancer, then it's better. Of course, what type of cells, et cetera, et cetera. So the goal is, can I segment those? A lot of histopathologists would like this, and we have a lot of tools for this. And what is normal, what's abnormal, what type of cancer, what stage. So first, but what I'm going to tell you first is automated pathology nuclei segmentation. Over the past, there's many different types of reasons why you want to deal with the nuclei, and why you want segment, and how do you do that with machine learning? So I'll say, I explain to you different methods, high level, and so you can appreciate what's happening. So the goal would like to achieve similar performance with much less annotation effort, similar performance to humans. So we would like to have weak annotations, because there's no way. One of the biggest problems that you guys have is, okay, you have a lot of data, right? But who's going to annotate so that I can use them for machine learning? There are methods where you can do weak annotations, as we say here, where you approximately say something about the data. You don't have to do full annotations. So what people have done, points, scribbles, bounding boxes, but you see they're expensive. Hard points are easier. And this is full mask. So for example, if you get this image and you do this, which is a little part of this whole histopathology image, well, can you segment all these cells? That's what I would like to do. So you have nuclei, can you see? You can see here. So at image level, you can see you can see points that corresponds to the nuclei, and then or scribbles people do, or bounding box. That's what typically they annotate with those that I collaborate. So we said, okay, what's better? So in terms of technology, there are all these problems. What data? How are you going to annotate? What model architecture? What neural net you're going to do? I mean, this is usually to us, but obviously we have to know your data and what you want, and of course, what's the loss function? What's the error based on which you retrain the neural net? So data annotation is laborious, domain knowledge is required, quickly supervised segmentation you like, so approximately. And the model architecture depends what you're doing. Are you doing CNNs? Are you doing UNets? Are you doing every 4 to 5 months there's a new model happen. That's why it's not anymore you just use a black box. You have to know the latest technology. That's why collaboration now is a must, because things are getting very complicated. I'll show you at the end what we do, so that you can input domain knowledge to the system, and I can use the domain knowledge that you know together with the data. So what's happening now, what you'll see as I go along, is you'll see that you cannot do data-driven only. You have to do data and knowledge-driven. That's where AGI goes. So I'll show you, we're about to submit next week this first paper, which I think is going to be a breakthrough for all of medicine and biology with this class of methods. So what we propose is a series of novel deep learning approaches to address multiple challenges of nuclear segmentation. It's just an example, right? It's not nuclear, it could be many other things. So we'll try to train with points annotation. So let's say somebody gives us points. You have the image, somebody has to give points where the nuclei are. So you do first what they call clustering to approximately find where the cells are, and then you do refinement. And together with another one is, K means clustering and Voronoi partition are similar, but what you do is you find areas now where the nuclei are, and then points and Voronoi edges, you compute those automatically. And then what happens is you have the input image, this is a UNet. This kind of network is not anymore like a CNN that goes like this, but you get the features from the low level and the high level. That's a UNet. It's being used in large language models or transformers, if you heard the terms. So this is probability maps, what happens during training again. So you do pre-processing on the data and you combine features, and then based on the loss functions, you train the whole system. So I'm not training only on images, but also on annotations. So I give both images and annotations together. Why UNet, you know, just in case you don't know, the reason why UNets are much better now than CNNs and this kind of approaches. The older approach that we say 10 years ago, which is like history for us, is because you couple low-level features and high-level features. The neural net, as it goes, first with the filtering it does, first finds low-level correlations between the pixels, and then slowly higher levels. We visualize neural nets on faces, and you see initially it's points, and then it says finds the eyes, the nose, correlations between the two. The reason why they work much better than traditional methods, least squares, etc., that you do, is because the features they find are nonlinear. So there is overlap between your eyes and your nose. That's the trick. So what they do here, effectively, they find correlations between nuclei, okay, which a human cannot do. That's the thing. Once I developed a method, I'm going to talk to you because of general computer vision. If you want to explain this scene, right? So you have data, of course, for the images, and you have knowledge, you have vocabularies, where the input is not based on PCA and these linear methods, where things are orthogonal. What you do is you have overlap between the components of your basis. So you see, the trick is, how do you make a basis that's nonlinear? And that's what we found. And also I have a pattern on this where I visualize the manifold, let's say on faces. I showed you as I'm turning my head, I can track it, right? So, but how can you do that? That's why the overlap I was telling you, in manifold space, in parameter space, your face, the frontal and the sides have to have some overlap so you can traverse the manifold. So that's the trick. Despite what they say, oh, you know, they're not, they have to disentangle, etc. I don't believe in any of that. Well, the reason why they work is because there's overlap. It's nonlinear. And that's why you cannot prove things, because nonlinear mathematics, they don't exist. Proofs. So it's intuitive, but it's more closer to what humans do. If you think about, let's say, dogs and cats, they share features, but at the same time, they're different. So how do you do that? If in our manifolds, they were totally separate, how can you go from a cat to a dog? You can't. So this is what we're exploring here. That's why these units, what they do is they couple features from different levels. Okay, and this is during training, during testing, sorry. So what do you do, right? So I'm telling you a little bit more the technology so that you understand what it takes to do proper segmentation. And we have in Nature these papers, etc. But so the effects of two types of levels. So sometimes people do pixel level accuracy, which of course or better shape information, or Voronoi labels, right? So you can do pre-processing and find Voronoi diagrams and find regions like this, right? So the question is, what is better? So do you have Voronoi diagrams or clustering, or you loss function, basically your error? How do you compute it? You may want to compute based on different types of methods. So when you look at the image, basically, are you going to get based on pixels? Are you going to do it based on bigger regions, the whole cells, or the nuclei? How are you going to do it? So, anyway, so this shows you, given the original image, that's where the cancer is here, and you can see here that we can get them pretty good. And this is when you mix them, which is even better compared to one extreme or the other. Now, how you do that, that depends on your intuition and how you model, given the knowledge that you will tell us. Okay, that's why it has to be together. I mean, it has to be collaborative. So how do compare performance with fully supervised? What if somebody really went into the painstaking job and did everything? Well, when we do it, on our own, we can do almost as well. If you do full masks, basically we do as better. Basically, if you do the approximately with pixel or object level, we do better than if somebody outlines. You know why? Because again, what you're not giving us is the overlap. So if you do approximately some pixels or some Warner whether some pixels is better for the neural net than if you do exactly everything. So the extension is we select 5% representative hard nuclei to annotate masks, which is better than points and random selection, of course. So we have a very small, typically, that I need. I don't need too much. So basically points, mixture of points and regions, and these are segmentation results. But I'll go into more details. So compared to fully supervised, basically somebody really doing them, I can get with 10% of the points, 90% performance. While if I do all the points, I can get 95. So of course you can tell me maybe this 5% could be important, but imagine 10%, and then I can refine with other methods. So that's what typically is done with this method. So I don't need you to go and do all the annotations very carefully. Of course, you can do it for testing the method, but what we do is we do very few, 10% of the points. We do 60% faster. We just interviewed yesterday somebody where there was a big deal now in machine learning. How do you speed up in hardware all these methods? How do you utilize better the GPUs? Because when they sit idle, you pay electricity, but you don't use them. So if you do all points and masks, almost the same. So if you do points and masks, so if you put features together, you can do as good segmentation as any. So the model architecture. Okay, so we talked about the data. What data do I need from you? I definitely need annotations. I don't need for everything, only 10%, 5%, 10%. Okay, so but what model architecture? What's the neural net? Okay, initially we started around 2018, 19, fully connected, right? Why fully connected? The way they were developed, again, it was at Toronto, where LeCun, who's at NYU, came to be a postdoc of Hinton, and he introduced him. What it does is what we used to do in Vision. In Vision, if you had an image, what do? You apply filters at different scales, so you can capture features. So what these methods do is they capture features at thousands of scales that a human cannot do, and find correlations. That's what a filter does. A filter is a mask that you convolve in the image and you find correlations. And then at the end, of course, you find the results, right? So these are hyperparameters, as they call, which means you have to choose, how big is the size of your filter, what are you doing, etc. So of course, it has a huge memory problem because how many are you going to put? What GPUs are you going to use? All these are trade-offs, right? So it's not like I just take a model and I apply it. You have to take the model, make sure you have enough GPUs so that you can do it in the right time. If you use two very small filters, which we do initially at the first layers, then it's good, but you can't keep using small filters. You have to give bigger and bigger because you want to summarize so that at the end you have few features based on which you make decisions. That's how they work. So anyway, these are details on the feature, but we do what they call leaky ReLU. Have you heard about these terms? So yeah, this is within the network. You see, you would use a network, right? But what we do is we go into the network and we play with certain parameters. So The reason why, if I tell you historically, when I was a student and I used to program those, we had one input, one huge layer, and then the output. That proved to be doesn't work. Why? Because when you have one big layer, it has to be infinite to find all correlations. It's much better if you have deep networks with lots of correlations as you go. That's what the architecture everybody uses. Now, leaky ReLU is they said from biology, you have the dendrites, right, and the synapse, and then there's a nonlinear function. So they try to emulate that. So this function is what they call this leaky ReLU. So Hinton did his PhD at Edinburgh in related to biology, etc. So he was influenced and arguing consistently with his advisor, as he had told us, about what the future of computation. So he wanted to basically discover features and not do it by hand that we used to do at this, with the previous machine learning methods. So what happens is he tries not to look, he looks at the brain. Of course, this is from 5 million, meters away, because we do know that the neurons, when the synapse, at the synapse, the function is definitely nonlinear. It's not the same function all the time. Depends what you do. But for approximation in these neural nets, you use the same function. And this function is called leaky ReLU. Basically, it's a way when you have inputs to a node, how do you select the output? Okay, so anyway, that's the terminology they use. But these days, we do either leaky ReLU or ReLU. Basically, leaky ReLU means, anyway, I don't have to go into these details, but just so you know what they're trying to do, they do know we are much more complicated than this, but it's a good start. So there are different types of networks. Dense net is one type. Full net, meaning every node has connections from the previous layer to every other node. So what are you going to do, right? And are you going to do ReLU or leaky ReLU? Again, this is parameters you have to change. If you just go and use a network, it's not going to work for you. You have to play with these parameters. So leaky, of course, is preferred because a big problem that I have with neural nets is when you have the error here and you try to go back to train it, if at the end here you have a big error, as you go along, the gradients vanish. So you have to be careful what kind of activation functions they call you're using. So these are technical things. I just want to tell you, that if that's why it's important to collaborate, because there's so many details to make them work. And we have to go through all this because in my conference to publish, if I say I'm doing a new architecture, I have to explain what I did, why I did it, and that my grades obviously don't vanish, because then there's no impact. I don't train the network properly. So, and then the loss functions, right? So here is also very important. What is the loss? How do you compute the error? What do you say the error? Like a human says, okay, this nuclei is not there, right? So how do you do that? So I would like to make prediction consistent with the pixels in the same instance, right? So if you plot the values, the RGB values, you have a mountains showing, right? And you want to have zero variance. And if you do that and you map them into the image space, then you can see that this is what is computing, and this is where the real image is. So you see it's starting to work pretty good. And then you can further refine, etc. One thing about the loss functions I want to show you, and like an overview. So the variance, it's an approach based on distributions. What I want to say, how much the variance of the input and what the network is producing, how much they vary. Now, how can you compute this? Okay, so the variance, right? Take the means and normalize, etc. This is standard term. So, but we need more than that. So not just the variance between the mean and the variance of the appearance of the pixels that the network is producing. You want more than that. So what I want to do is add some terms to the loss function, so I can constrain much better what it means to be an error, right? So this is always going to be a big issue when, let's say, we collaborate. You tell me, okay, do segmentation. Now I have to think, what does it mean to segment properly? Is it the appearance? Is it the variance? Is it the combination of those? What is it? Okay, so this is issues. Okay, so so this shows you, let's say, a human gland, right, with large lumen, for example. So I would like to segment it. Okay, so this is the ground truth. So you see, we're pretty good by doing this approaches. So if you do a full neural net, fully connected, you get this kind of on the same organ test set. I use publicly available data sets, right? Because they've been developed and they're publicly available because you won't test new methods, right? Or there are different organ test sets. These are different publicly available. They're for related to lungs, to all sorts of cancers. If you do the variance between the pixels, not only the pixel, but how much it varies with the surroundings as the error, you do much better, as you can see here. These are scores, again, technical, dye scores, AGI. I don't know are you familiar with those? So these are very technical terms of how we measure things. How we measure the actual values, how you measure the distance between the values, these kind of things. So we have the state of the art with this kind of methods, and this is our approach here. You can see the nuclei here, and then the cells in the nuclei here, and you can see that we can do pretty good. There are some cases when you do 2D. I'm looking forward to somebody giving 3D data, so you have slices. So because sometimes when you see, you can see here their overlap, but sometimes we can see the overlap here. It's not bad. You can see there are two separate ones. But if I could see in 3D, because my experience is that when you do in 3D, it depends how you slice. There could be fake overlaps or fake one, and you can't do anything about it. So anyway, so this is the state of the art in segmenting. And I've segmented many other things where I want to find the cell and the nucleus, depending on the cancer. The nucleus is at the border of because of cancer, at the border of the cell. It's at the membrane, etc. So they could be all over the place. So anyway, with our methods, what we're trying to do is analyze automatically the whole image and find the cancers. And then you can measure, given the cancer, the size, etc., statistically. So there are issues with over-segmentation. If you're not careful, sometimes things get overlapped. And basically, your filters in your network miss the point. They don't have the resolution to see the boundaries. So that's another issue we have to worry about. Now, let me tell you something which is probably more interesting to you. Okay, this is more the technology. I want to show you that there's so many different networks I may use depending on the application. That means you have to know the architectures. And within the architecture, you have to decide how you're going to model the layers, et cetera, et cetera. These are hyperparameters that require knowledge. And of course, I can explain to you, I mean, if anybody's interested to collaborate, is to tell you why we're doing what we're doing. Okay. Now, what if I wanted to do genetic mutation biological pathway prediction? What if somebody basically shows me, this was a paper we had in Nature Precision Oncology. So this was collaboration with some people from Stanford and some people actually from here who went to Yale. But the thing is, they gave us data sets, and what we're trying to do is, given an image, right, high resolution pathology scans, right, I want to find the pathways. What's a possible pathway? There are many pathways. I mean, I don't have to explain this to you. You know that much better than I do. And I would like to be able to do treatment insight, prognosis, biomarker discovery. Can I discover cancer earlier? Okay, because the networks can see correlations here that the eye cannot see. Okay, and of course detect the molecular signatures because this is important for cancer, right? And these days I'll show you, given an image, I'm going to produce to you text what's the problem is, so that you understand. So so let's start with this. Okay, so genetic is required to find the targets. It's not routinely used, time consuming, expensive. More biomarkers are needed, different types of cancer. I mean, the problem, as I understand, is the beginning. I mean, what they're looking at least what they were telling me at Stanford and the other places, they want to detect cancer early. And what genes contributed, okay, or pathways. Can you find this? So I would like to predict gene mutation and pathways from histopathology images better than a human, which I think we can. So histopathology are now ubiquitously available, of course, and we want to explore genotype-phenotype relationships. It's beginning to be a very hot area in my field, meaning to develop new methods. And actually, they contacted me from Gladstone Labs. I don't know, are you familiar with those? So I know the head of it because I organize every year a histopathology analytics workshop next to me, to CVPR. Just so you know, CVPR, which are organized in Denver in 2026, is the highest impact in computer science. The impact factor is over 60. Okay, so it's better to publish in that than a journal, because 60 impact factor is very high, right? There are 15,000 participants, which project to go to 20,000. So this field now is, and then all the big companies like Google, et cetera, who are trying to do analytics go there. So increasingly, I've been doing this work for 10 years. We started with 200 people, now it's going to 1,000 people. It's a workshop, just a workshop. Okay, and then everybody comes from all the big labs who do biology or histopathology, because they really want to do exactly this. Okay, so I think it's very important that we collaborate, because you know, everybody else is doing it, especially UCSF with Stanford. They're trying, but they failed because they were fighting. So now they prefer to work with me than to work with, you know, these are politics of within biology. But anyway, so so want to explore this thing, right? So we have different, of course, data sets. So whole slide images with labels from the Cancer Genome Atlas. You're familiar, right, with this? So, and here you have, of course, the protein data bank, but this is for cancer. So what we can do now is take data and then do analytics, right? So they have 10,000 tumors, 33 cancer types. So there's a lot of companies slash the big universities have a lot of resources to make publicly available. Is to their benefit if we develop better methods, right? So they can make their life easier and do discovery. So so model training and evaluation. So what happens? You have patients, right? You have a training set, a validation and testing. I don't know if you heard about, when you do neural nets, this is very, very important. You train, okay? But you train on data, but then there's another step called validation. Have you heard about this? If you don't do validation, it means you're overfitting and your testing will not be good. So you don't want to overfit your data. You don't want to fit exactly. You want to approximate your data. And with validation, you change your hyperparameters, and then when you test, you get good results. This is very key into learning. So, so what are you going to learn? You can learn graphs. Because why graphs? Why I can learn based on graphs? Because I'm able to make a decision given an image from different areas. The image, not just the vanilla thing. I take the whole thing, and that's black box. We don't want that. We want to know which area contributed the most and why. So, for example, we do find TP3 is a tumor suppressor gene, cell proliferation, cell survival, and genomic integrity, and mutated frequently in breast cancer. So if you're into breast cancer from this data, can we find those? So we can do that. The area under the curve is 95%. So we can discover this tumor set, this gene, right? So this pathway, sorry. And this gene, right, TP53. So there are a lot of genes. I learned those from the people I was interacting. There's another one, FGR1. So amplification, cell proliferation, differentiation, most frequent genomic aberration in breast cancer. So there are all these genes, right? Well, imagine what will happen if somebody with hereditary, with bad hereditary genes goes and does a testing, and maybe we can find, because the biggest problem I do know, because we have relatives with breast cancer, is when you look at it with CT or MR ultrasound, and at Penn we had designed actually a coil for the breast to do better MRI than general MRI. Can you detect it earlier, the cancer? And you can with these methods, right? So if you've trained it to find correlations between different areas of the image, then you can do much better detection of these genes. So and some of them there are different pathways. I mean, that's the very bad cancers where you have 2-3 pathways in the cancer. So that's another one, P53, with more aggressive disease. Again, there is, better. Of course, there's milder cancers, aggressive cancers. How do you find those? Can you find the genes? Can you predict? You should be able. If we have basically ground truth from the data, and you have the data itself, we should be able to find the correlation. So there's another pathway, regulator for cell growth and proliferation, another basically pathway for cancer subtypes. So there are different types, and we can find those. So for example, you have all these tiles, right? So I can find the A is the tumor tiles, right? These are all tiles. T53, mutation. P53 pathway activity here, top 20 weighted tiles. So if you give me a lot of tiles, can you give me the most probable that they have this pathway? And these days, I want to also say why, in your terminology, and I'll show you what we're doing now. So basically, this is what it is, and this is the pathway. I mean, this is diagrams from biologists, obviously. But what I want is, given those, if I know with some ground truth that they exist, can I automatically figure it out? Another one is cell cycle pathway. That's another one. This is highlighted, top 20 weighted tiles with cell cycle pathway, okay? So this is the cell cycle pathway. So you can find here, examples of these tiles, given thousands or millions that they give us. And anyway, these are the images we get. So in breast cancer, the CNN, we use the CNN fully connected. Point mutations of 6 genes, we tried. Copy number alteration, another 6 genes. This was in a Nature paper, okay? So we could find better than humans. And not only that, but it shows that we can discover things that didn't exist. 3 pathways from mRNA expression, 2 pathways from copy number alteration, visualization of weight maps we can find, basically how important are scenarios, how sure we are. Similar related in related gene and pathway results, and provide new insights to find potential biomarkers for targeted therapy. So this is an area that's growing so fast that you can't believe it, how the technology for these methods. How about more recent, what do we doing that? We would like to do pan cancer. We just submitted a paper about pan segmentation. It was just accepted into, I'm not going to talk about this, there's too many technical details, but we can do segment anything in the body from data. All the structures, very difficult. No doctor can do it. They specialize years and years, for certain parts. Also, another thing I won't tell you is like, if you show a doctor a random image without telling from where, they don't know what it is. There are many areas in the brain, like the brain, and for example, the abdomen have very similar appearance from MRI or whatever. But this is, of course, about, biological images. So pan-cancer image-based detection. So there is this which has appeared. So there's a big effort. How can I segment now and detect any type of cancer from histopathology? What we do is we call what they call graph-based approaches. Have you heard about those? So it's not anymore CNNs. This is, I'm doing different regions, I'm creating a graph based on which I make assessments. So captures intratumoral spatial information that's beyond a CNN. CNN is a thing of the past in most ways now, because they use transformers or graph methods. So what I'm trying to do, I'm trying to make a prediction about the tumor, right? Where's the cancer? And I'm trying to make a graph from different areas, okay? So I want to enable special characteristic representation and measurement of tumor types. And that's a lot better because now I'm getting votes into my assessment from many areas. So what do we do, right? Given a histopathological image, right? And we do this tumor detection and path selection. This is what we're trying to do. But how do you do that? So you have the graph, then you do k-nearest fully connected graph. So we'll, instead of one input or an image, we convert that into a graph. And from this graph, we're trying to find areas where there's different types. So basically, within this graph, this area, the subtype classification, etc. So what we're trying to do is convert the image into a graph. Then the graph, we prune it, and we find the areas where the cancer is. Okay, so that's what these methods do, this graph-based. So, we constructed the spatial graphs. So basically, the image is a bunch of pixels. From the pixels, you find areas, you create a graph. So you construct the graph. GNN is graphical neural networks, molecular profile prediction, and then this is what it is, gene mutation, and all these pathways like CNA, mSI protein, what's wrong with it? So it becomes a lot more, if you want, detailed compared to a CNN. So extract specially connected features from tumor titles to advanced image molecular analysis, let's say in this case, colon cancer. So it doesn't matter to me what the cancer is, as long as I know, of course, I have some ground truth, and then I automatically create these graphs of connectivity and appearance of the cancer. So how about histopathology key mutations? So for example, we looked at the following, right? So it starts in the intestinal epithelial cells. Then you have this ABC mutation here, pathway, which creates the aberrant crypt foci. And then there's another pathway here, which can become adenoma and then carcinoma. Okay, So we want to seek to build image-based graphs and identify detectable evidence for cancer molecular outcomes with therapy implications. So we don't want anymore to go and see, at MRI, etc. You go to histopathology, and I'm trying to find the gene. Can I find the correlation? Instead of a pathologist doing this, can we find it automatically and help them, right? Not to replace them, obviously. So what you do is you have special graphs at various levels from various images. And what you do is you fuse them. That's the output. Basically, you have lots of images, lots of graphs or histopathology images from people. And then what you do is you create these graphs and then you fuse them. You see here feature enhancements. This is what I was telling you before, like in CNNs. You take some features from lower layers and you use them also in higher layers and infuse them. I mean, this is called feature enhancement methods. So anyway, this is the idea here. So what did we do with this one? So let's say you have an image here, histopathology, and what you do is you would like to find in red, yellow, and green, you want to find the areas where there are different mutations, right? So the top in this color, which is this color, is to identify tiles from subgraph model 1 within the purple region. In red, you find top 10 identified tiles from subgraph within the region. So you see with cancer, right? So these are region color correspond to results from each subgraph model to the right. So basically, we create a graph, and then the graphs are from different regions. We said before we put them together, right? So by doing that, then you can do much better identification of what it is, what the problem is. And and what you can do if you do this fusion, your AOC, of course, AUC goes much higher, goes to 90%, 95%. So we can detect with very high accuracy compared to previous methods, or, we're not doing graph composition, basically what we call the ensemble model. Much better detection of what kind of cancer, what are the genes. Okay, all this we published in Nature. But I want to tell you now, okay, what's happening, right? So clinical and preclinical perspective, like, so you need high quality multimodal data collection. There's a huge effort to do not just one type of data, but multiple. And with annotations, of course. Improve disease understanding, diagnosis, and outcome prediction. Based on what I showed you, I can detect pathways, right, from images much better than a lot of pathologists. When they get tired, I know because I watch them. In the morning, they're very good, and they miss cancers. A lot of problems there. So treatment response prediction, prospective clinical trials. I mean, imagine with these methods to run them, because sometimes we use FPGAs, you're talking milliseconds. If you take a person to read these things, it takes hours, right? So imagine what can happen, right? That's why on May 14, Steve will come, because I have this IUCRC, and Merck is coming. Finally, I think they realize that we can do analysis for them, we can do clinical trials technology, right? So Sanofi Aventis, I used to work with those when I was at Penn. So I think they're coming back so that we can do analysis, because they have huge needs for discovery and analysis. From a data perspective, high quality data augmentation and annotation. If you give me data, I will augment them for you. Statistically, basically, I'll make statistically similar, but augment them so I can have more samples. Basically, shift them, rotate. There's a method called data augmentation. Robust feature extraction development, of course. If you have the data, long tail data handling, sparse data, often imbalanced, multimodal data, and of course, large data. The protein data bank that you have here is a great example. We can augment statistically. We can do a lot of things to do better analytics. Now, you can also, guess 3D structure. We do, from images, I can tell you 3D structure of people, right? So you can do a lot of things. And we can also encode PDs, like all the stuff you used to do with physics. I can emulate with computers. I mean, this is another talk to give you, but so what is the opportunities? And then I'll show you the newest method I'm very proud, because I think it will be very useful to you. So no more we call AI, we call AGI perspective, right? Models are able to deal with long tail data, very important. Out of distribution. This is called, when you do learning, out of distribution. Like I give you this data, but can it work on data that's not statistically similar? Statistically means transfer learning, if you heard the term. But I'm talking out of distribution. And multimodal data has to be, if you want to do really clinical or even preclinical, the more data you have or the more knowledge you have to do that. This is, I don't know if you have you heard about LoRa? So in large language models, they're closed models, right? What do we do, right? So I don't know what they've been trained on, but I can change the weights a little bit. So these are methods to speed it up, the weight training. It's called LoRa. Large machine learning methods to couple pathology with clinical and omics data. The future will have a lot of data. Of course, if I'd learned properly all this, from pathology, I can predict the omics. I can predict a lot of things. And I say, look, what's this guy? Because, probably he has this pathway. And they can check. Distributed learning, anyway, these are more learning methods, federated learning. If your data is at different locations that cannot leave, then have patterns for federated learning. Generative, when I say federated, these are large language models, but federated. And I have patterns. Anyway, and the big thing is explainable. Okay, what am I doing there? Okay, let me show you this. This is a paper we're submitting next week to MICAI, MICCAI that you heard, is the best medical image analysis conference I've organized in New York. I mean, it's a very good conference for technology, though. So what is the problem, right? So medical images or preclinical play a vital role in clinical biological workflow, right? Precise diagnosis, non-invasive viewing, early detection, et cetera, et cetera. AI is revolutionized as we speak, and it's going to intensify. Rapid processing, you can do much faster, consistent, enhanced accuracy, low inter-observer variance. We used to have NIH, you're very familiar, we have three doctors delineating boundaries of the heart, they all differ. We don't have that. And of course, automated interpretation, but as a tool. I don't want to replace anybody. I want to give them better tools so they can do their jobs better. And of course, the biggest challenge is the black box. Like I showed you all these things. If I show you the details of what we do, I mean, you'll be overwhelmed, right? Because you're from a different discipline and you shouldn't have to know. Therefore, you have to trust it, right? But it's a black box. You would never trust. You're a human. And I wouldn't trust if I didn't know these neural nets. So how can we obviously, you need a new approach. As what happening is a new revolution happening. Big companies, new architectures. You know why? Because, I mean, the little I know about the brain, the way we think, we have a low memory. And it's not like these architectures we have. You do processing and you index to memory very fast, okay? And you don't retrain your system every time something new happens, right? So something has to change. So here's an example of what we're trying to do, right? So I want to use domain knowledge. I want to know your expertise, right? From the data, you can tell the expertise. it's taken us 2,000 years to go to be where we are, right? But why not use domain specific database criteria, images, blood, genomic? I want to use many data. Other types of knowledge, types of disease severity, outcomes, correlations. These are when somebody practices, or you as biologists, you know a lot of biology, right? You don't. That's how you filter your data. There are three terms that we will hear: grounding, alignment, structurability. Grounding means I want to make concepts from the data the way you understand as a human. Of course, the internal representation will be very technical, but what it outputs should output, okay. Alignment means the decision making is human-like, okay? And it's open, it's transparent. And instructability is if you don't like something, you should be able to tell the network, I don't like this, but the way a human would do it. Not to have to delineate very carefully. So can you do that? So yes, we can do that. So I'll give you a case of melanoma, right? So the human knowledge says you have to have a color, it's mixture of pigmentation, including but not limited, etc. That's how they practice. Shape, border, pattern, texture. These are all examples. I remember when I first did breast cancer detection at Penn, I used from some doctors, what they were looking to create myself features. At the time, there was no neural nets working. So now here's the thing. Here's the new method. So I want to encode the visual information from the images. I create an image feature vector. And I want to couple this, associate with the human knowledge. So how do you do that? We change what they call in transformers the attention mechanism. I mean, it's fairly complicated, but what it does, it tries to find correlations between, I'm translating the representation of the network, which is unintelligible to human, to features that the human understands. And I'll show you in the next thing that I can find now probabilities about color, shape, border, pattern, texture to what a doctor or a human knows. And then I do a matching between the visual parameters here and the text, and I create a prediction. And the prediction is the cancer, or what is the cancer, and why? Because I saw the shape, etc. So imagine if you in biology, you tell me, okay, here's the images, and I know these things. Try to find correlations. Okay, not by eye, but automatically much better. So here's what we do, right? So what we do is I find out correlations between the text, the embedded knowledge means I go into the space of the neural net to understand the text. I transform it, and I transform the image features in a space that I can then correlate. So that's the method we have, and I think I should patent this because I think that's going to become AI. Now I output and I say it's 92% melanoma. Why? Because I see 87% this color, this color, this color, this color. Now you can understand. Okay, so this is where it's going. There's going to be a lot bigger revolutions happening, not only by my group but many others. And I know because Google asked me, they are really looking into new architectures in New York because they know that cannot be black box. If you want to go to the next level, it has to be explainable. So this is explainable. But the difference is, and these are large language models, that's the way I encourage large. So large language models, if you just use them, it will give you garbage. But if you couple them with images and knowledge, this additional knowledge I give to the large language models, then it will work. They don't work for medical by themselves. They're not trained for the specific knowledge. So we need to add knowledge, but you need to correlate. And by the way, I see visual here. I can have blood tests. I can have many other things. I can correlate. So it could be multimodal, right? This is the first step I'm doing. So if you see what happens, we're getting way better. I mean, way better. 0.5% is significant for these data sets. These are open data sets. The dermoscopic skin lesion images. 7 disease states. These are open data sets that I have to use, so I don't have any other data sets. Test rate, etc. So the method is interpretable. The method outperforms supervised learning. So if you try to do CNNs with exact supervision, we do better. And I'm not surprised. I'll tell you why. Because I find statistical correlations with what exists, but not being exactly saying this and this and this, because a human may be wrong. So in the future, I want to introduce additional knowledge, develop improved knowledge representation alignment. Right now, if you see what I do, we do separately color, of course, the neural net correlates those in a non-linear way. But what if it's a graph to make a decision? Basically, I saw this, then I see this, then I see this, the way a smart human does, right? Not for this case, but for other cases. So it's called graph. So I can create this correlation to a graph. So I think the sky is going to be the limit. So wait a few years, your jaw will drop what these things can do. I used to believe initially in the 90s, they don't work, but the moment we did the ReLU, this nonlinearity, which resembles the human neural nets, even though it's far from them. But now this is trying to impose memory and knowledge, right? So it finds correlations given data, and it doesn't care how many data you have or different types. Okay, so but this is I think what's going to be. So so we're doing more. So you see here we get more than 2%. Like it's on this data set, and we haven't published. We're perfecting the method. So it looks like it's working better than other methods. So if I want to conclude, we are developing novel deep learning methods and AI, right? So we provide efficient and effective means to analyze nuclei tissues in pathology, what I showed you, but not only. Your field or other clinical fields. To me, I want to make methods that are agnostic to what I'm doing, right? But they're general purpose. Of course, they need tuning given the application. So multimodal analysis has to be pathology, omics, clinical data. If you want to detect cancer early because curable, you have to use this kind of methods. Promise to uncover new insights. And what I want is like in the Nature paper, we did find a correlation between pathology and cancer they didn't know. So you can find new things. Like I don't want to just regurgitate what you guys know. I want to do more than what you know to help you find even more correlations in your research, but never to replace you. I mean, that's the big mistake. If you hear some people saying, I'll replace, no, we're not there yet. We're not, doesn't do creativity. Creativity means I have one distribution, I have another. So creativity means your knowledge is in your brain. You haven't correlated. Right? If you see the history of Einstein, I know well physics, there was the examples of that space-time bends, right? Except they thought classical mechanics, nothing bends, right? Well, he said it bends, right? So I'm sure when you see things, information is there. You just haven't thought about it. So creativity is, mathematically speaking, you have two distributions that are uncorrelated, and you correlate them, you find a new distribution. That's a new thing, right? A new distribution. Well, it doesn't do that yet. Well, maybe someday we'll find a way to do it. That's why I'm saying it's not human. It's within distribution, or at least with this explainability, I know if I see something, if it does belong in my distribution, or I say, look, I don't know this. So let the human help me. Okay, that's the extractability that I was talking about. So I do want eventually to be able to get discoveries. Computational pathology is evolving rapidly given recent progress in these methods. And of course, large AI models, what they do is the way we evolved, if you think about it, the world has all the details in an image, but you speak to each other abstractly, right? If you give a description of a scene, I cannot reproduce the string. It's abstraction. If you think of a bathroom, You think of the sink, the mirror, right? The bathtub and the toilet. They may not have all of this, but you don't think about what's the tile or what's the color of the tiles, right? So you cannot reproduce by description of a bathroom exactly the material. You have to see it, right? Or feel it with your hands. So combining language, which gives you the abstraction and the big picture, with other data helps. That's what we're doing. Okay, so that's how AI works. And that's why when we evolved, we were not speaking initially. It's very clear that we first started gesturing. But gesturing, you have to see each other, right? And sound is less energy, and you don't have to see each other visually. So, and it's very clear in the brain. I don't know if you've seen the structure of the brain. It's very clear the new brain and the old brain. So very clearly all this. So therefore, and we're multimodal by definition. We see, we feel, we smell, right? How can you do all these things, right? And we can now with language models. So sometimes they call them foundation because they're pre-trained. Combine multimodal data with domain knowledge. This is what I showed you. This is what's happening, I think now. This is the hottest area here. To improve efficiency, validation, robustness, and disease understanding. Very important. If I can give you better statistics of the appearance of disease, then we win big time. Right now, I'm have an R01 with NYU on cardiac. Has anybody thought about how many data sets they've done statistically to tell you what's normal heart and what's an abnormal? No more than 100, because they couldn't 50 years ago. And do you know, with normal heart, do you know the variation is huge? 400 diseases in the heart, 400. No doctor can diagnose properly. Do you know what they say? Asymptomatic heart. He died because asymptomatic heart. What is this? It means you don't know, right? And you can measure. So we can measure now. We're submitting a paper for the first time for desynchrony, where they put pacemakers. So I can tell you, by the way, all the companies are fighting us, but we're publishing it. If you put a pacemaker, 30% it works better. 30% is the same, and 30% worse, makes it worse situation. Well, they don't want to. I asked for my NYU to give us data, they don't want. Only a doctor I work there is sneakily giving data because they are paid by the companies. This is $1 billion industry, right? So you understand, if you want to really push the envelope of science, you need these methods. And we find correlations between effectiveness of a pacemaker and types of desynchrony, which they don't know. So there's a lot of things we don't know, and we can use these tools to find them. And that's why I'm very excited with these methods. And of course, this is what's happening, the general intelligence, right? Artificial general intelligence. This is the new thing, right? AI is associated with machine learning, with black boxes. No, we're going beyond that, towards closer to how humans operate, even though the underlying is, of course, computational, etc. So I can see applications everywhere. And as I said, this method, I think, will have a big impact. And I want to thank, of course, these are all my students who are now in big companies. Actually, my first student was female. She's now vice dean at Penn State. So I'm very proud of her. I mean, I don't mention her, but I have a lot of PhD students and, some in this area, in important areas. And she's also in medical. The way another guy is in Delaware and Stony Brook, etc. So this area, I think, when in collaboration with other fields, I think it's very lucrative scientific outcomes, and I think will help improve our lives, I think, big time. Yeah. So I'd like to.
A great note on which to end, improving lives. Please join me in thanking Dimitris. So you've gone way over time, which was your prerogative, of course. But so we won't have a Q&A. But what I did want to say is that I'm very optimistic based on
Yeah.
the picture of the future that you're painting, that we'll be able to combine the expert biocuration in the protein data bank with this AGI approach. And the other area where I'm hoping you're going to have an impact more quickly is in the setting of immune checkpoint therapy for cancer. So right now, if you watch the ads on TV, you'll see that Bristol Myers Squibb is advertising two antibodies that hit different parts of the immune checkpoint system, with the goal of trying to treat successfully cancer and not just treat it but cure it. Jimmy Carter, for example, was one of the beneficiaries of immune checkpoint therapy, and he's going to die at probably age greater than 100 of something other than the melanoma that he had that was in his brain. So right now, the therapy that Bristol Myers Squibb advertises, two antibodies in combination, has about a 50% effectiveness rate. But we have no idea why 50% of the patients don't respond. And combining all that information, responders versus non-responders, full genetic profile, et cetera, proteomics, et cetera, I think will hopefully tell us why, and then approaches to those non-responding patients can be fine-tuned to improve outcomes. So thank you again.
Thank you very much.
It's a brave new world.
Yes.